Good morning, all of the participants. Welcome you all on the third day of National Webinar Series 2020. Today, the 23rd August 2020, the third day of the webinar. First of all, once again, I welcome you. And to give up, yesterday, the video lecture of Dr. M.C. Agarwal, a lecture which was very, very important, giving the directions to all the parasitologists, maybe young, maybe experienced, regarding the working in different aspects of the parasite. He not only opened many ways, many directions, but he also gave good points to how to work and go to the problems of the farmers. Later on, Dr. Shri Kumar gave his lecture on the wildlife parasite. Very, very few parasitologists in India, they work on wildlife parasitology, but area which is not completely explored, which has a sea of things to be studied. So that was a very important lecture. Afterwards, in the equine parasitologist, we have the lecture of Dr. Sanjay Kumar, a research scientist at National Research Center Equines. He gave on the equine pyroplasmosis, especially he emphasized on Thalaria equine, giving many ways of its diagnosis, many perspectives of its control, as well as many ways or many kits which are likely to come during the days. The 23rd August 2020, we will be having two lectures. One of Dr. Rajesh Katoj, a very important, very learned, very hardworking head of department of Jammu. In addition, he is also holding the post of Dean Student Welfare. He will be talking about the herbal ectoparasiticides. After that, we have the second lecture of principal scientist at IVRI, Dr. Rajat Garg, and he will be talking on coccidiosis, a very important disease, and or you'll be finding in poultry birds. But before these two, we have a video of Dr. M.B. Chabla. Talking about the Dr. M.B. Chabla, he is a person who is ex-head of department from this very institute, Department of Veterinary Parasitology. And it was the Lajpat University of Parasites. Very important, very esteemed researchers, as well as a very, very well excellent teacher. Teaching skills, his expressions, it is beyond any comparison. Talking about his research skills, we go for the coccidiosis of the birds, or coccidiosis of the buffaloes. We always see Amelia varelli, and it is for your importance, for your knowledge, that Amelia varelli in buffaloes at Karnal was discovered by Dr. Chhabra. The same was also published in an international journal. The pioneer who pointed out the association of Toxoplasma gondii with caprine abortion. Again, a very important work carried out in 1968, published in an international journal. In addition, he has guided one of his doctoral students who later on happened to win the ICR Jawaharlal Nehru Award. In addition, during his total services, he has published more than 170 research papers, out of which 1/3 of them were in international journals, and 1/3 they are the review articles. Even after retirement, he took the premature retirement few months before, and he has a distinction to work as a full-time consultant in a pharmacological industry post-retirement. So just before the retirement, after 6 months before the retirement, he, not only for this, even after retirement, he's working on books, and the last book which is the parasitic zoonosis in the new millennium, was written and published by him in 2015, almost 22-25 years after. Yet, he is very, very energetic, very, very enthusiastic, and fortunate. We are fortunate to have a message of it. So we'll be playing that video message.
Hello friends, it is great that the Department of Parasitology is working in our. To an old timer who mentions the virtual thing usually means home delivery of knowledge. Technology is a great means of information dissemination, access and dissemination. Like this, webinar is not only the new norm, but also a compulsion of the COVID times. I wish to congratulate the organizers for this wonderful initiative, which, besides the objective of advancement of science, helps in certain ways to lessen the prevailing pessimism and negativity of the COVID-19 for all concerned. The theme of webinar is highly relevant and inclusive. In my time, the field of veterinary parasitology was held hostage by zoologists with their misplaced emphasis on systematics for classification, morphology, speciation, etc. We vets are more concerned with the harm caused by the parasites and with ways and means to prevent or alleviate the suffering and distress. As such, the priorities of the webinar, today's webinar are on Dr. Sukhdeep Kaur, research credit for working in a galaxy of the technical program covers a wide spectrum of topics. I firmly believe that hygienic measures and nutritious diet are just as this particularly the dietary part has led to coining of the term nutraceuticals, which our poultry Coccidiosis friends may well understand. I'm also happy to find wildlife parasitism being addressed in this seminar. With increasing interface between humans and wild animals, opportunities for transmissions have increased lately and an upswing, notably among in the viruses that have occurred lately, SARS, Ebola, and lately COVID-19 are good examples of the precipitating results of this environments. Herbal agents for control of our parasites should in fact form part of an integrated sustainable control strategy. In the current era of rapid generation of news and technology, it is important to have such meetings. The possibility of incorporating lab demos of new procedures should be explored in future meetings. Prerequisite for good teachers is to remain students for life. Learning is an endless process. The webinar is a knowledge sharing and dissemination. For such event is always and responsibility, but I am sure that the dynamic Dr. and his young team rise up to the occasion and will deliver their best as professionals will ensure widest participation on their part. This opportunity, which gives webinar a grand success. I end with thanks for one and all.
Thank you, sir. I, along with my young team, as per Dr. Chawla, thanks Dr. M.P. Chawla for this wonderful message. Now I invite Dr. Viswaranjan Maharana to continue the lecture series. Thank you.
All are requested to close their camera. Please put off their camera, otherwise we will be forced to remove. Dr. Ananda, kindly put off your camera. Dr. Anand J, kindly put off your camera.
Everyone. A pleasant morning to one and all. Thank you for joining in the Day-3 webinar series. Just a little housekeeping before we get started. If you have seen the presentation, please type in the chat box. We will be answering those questions at the end of each session. On behalf of the organizing committee, it is my pleasure to welcome our today's speaker, Dr. Rajesh, Dean Student Welfare and Head of the Department of Veterinary Parasitology, Seri Kashmir University of Agricultural Science and Technology, Jammu. Dr. Kotos is presently acting as DSW Head Department of Veterinary Parasitology. He is a fellow of IAVP. He was awarded Ismail Oreson Award and Srimat Nisamani Parija Award for his valuable contribution in the field of parasitizoonosis. He is a recipient of the Dubey Young Scientist Award. He backed 14 projects from various national and international funding agencies like GST, RTVI, NABARD, etc. Now 3 projects are currently running under his leadership. He successfully guided 8 MBSc and 8 PhD students. He has published more than 60 research papers in national and international journals. He has also published 4 books and many book chapters and lead papers. During his long 30 years of service, he has worked in some of the prestigious veterinary colleges of our country like LUVAS Mathura, Palanpur, and Scotch J, and got a chance to work with stalwarts in parasitology like Dr. P.P.S. Chauhan, Dr. H.L. Shah, and Dr. A.K. Misra. Now, without any, I would like to invite Dr. Kotos sir, our first eminent speaker, to go ahead with his speech. Over to Dr. Kotos sir.
Presenting my talk. It's a basic talk what I am talking about, because I'll be talking about herbal insecticidals or acaricidals where majority of the scientists are working, and even the whole world is looking towards herbal compounds. Why this need is there? Emergence of resistance is the major problem, because we have limited group, 5 or 7 groups are there, and we can use those groups to control these arthropods. But slowly and slowly resistance has been reported all over the world. That's why we are heading towards herbal side. All of the world is looking toward the herbal side. And the important point about these chemical insecticidals, they have a residual effect on the animal food products. Remember that we are very clear that DDT came into food chain. We have problem with the rotenone. Even nicotine also is creating problem. But in spite of that, the chemicals pyrethroids are little safer because we are using in good night, which we are keeping to keep away the mosquitoes from our houses. And then these chemicals also cause environmental pollution because they are degraded very slowly, particularly organophosphate compounds. They are degraded slowly. They have an advantage over DDT, but they were degraded slowly. That's why we went for the other group, pyrethroids. So other thing is that undesirable effect on non-targeted species is an important character of these chemicals. Because of that, certain arthropods, which are of immense importance, because as you know, arthropods accounts for 80% of the animal kingdom, and they are the main pollinators. Once they are not there in the nature, the diversity will reduce, and that will be the end of the world. and they came onto the earth 400 million years before man came onto the earth. So they are well adapted in this system. So everybody is looking towards herbal products. If we look into the world, herbal medicine market value to surpass 129 USD billion mark by 23, and it is growing at a very fast pace. Compound annual growth rate is around about 5.88%. And if we see, there are four major categories. One is herbal pharmaceuticals, another is herbal functional foods, herbal dietary supplements, and herbal beauty products. But we are concerned with mainly herbal pharmaceuticals, and it is the major part among all the four. It is accounting for 50.9 billion in 2017 USD dollars, US dollars. This data is, and herbal, by which particular product is being shared the most. There are tablets, there are powders, but it is the extract which accounts for the largest share. It is approximately 23.7 billion in 2000 billion, CGR in 16.5%. If we see that the share in ectoparasiticidals, it is hello. Share in ectoparasiticidal is approximately 12 billion USD dollars. So why we want these herbal insecticides? Because they have a narrow spectrum. The arthropod which we are selecting to kill, or we want to reduce it in the nature, for that particular we can prepare that particular herbal insecticide. Impact on insect is gradual. It is not very fast. As we see in the chemical insecticides, there is a knockdown effect. But in this case, the impact is very gradual, and around about 90%, round about 90%, 90%. Hello, hello. Impact on insects is gradual, and 90% of the insects, they are lost by this applying by this chemical, but 10% exist. These chemicals are economical, because if we see economics, because generally we select a herbal insecticide, particularly from the weeds, and we generally select a part of the plant which is easily available. Particularly leaves are majority cases they are being used for making herbal insecticides. We use seeds also, but in certain cases roots have also been used, but that we use less because that destroys the plant. So the other important thing is no report of resistance. There is we don't have any report regarding resistance in against the herbal compounds. And even if certain chemical resistance is there, that particular strain also when herbal insecticide is applied is it acts on that also. So that is an important point here. And biodegradable, very easily they are biodegradable and safe to mammals, fish, birds. That's the important point. The important point about the herbals, they act either on through juvenile hormones or chitin inhibitors. I'll be explaining this in the next slides. So if we see herbal insecticidals, there are around about 1,005 plants with insecticidal properties and 384 with the anti-feedant properties. 297 approximately with the repellent properties, which I got from the literature. This is mere reviewing of the literature only. Remember that repellent doesn't kill but make the host unattractive. While there are rosemary, cedarwood oils, all are being there. 31 plants are there with growth inhibiting properties. Neem and essential oils are there. 27 attractant properties. That is very important. You know, we must exploit the attractant property also. Lure the insect into the trap where it is where it can be killed by contact poisons. If we see, it is a co-evolutionary relationship of allomons and karyomons. Allomons, any chemical substance which is produced and released by an individual of species that affects the behavior of member of another species to benefit the originator. Actually, it happened like that. So, certain plant develop a substance or chemical or smell which doesn't, which was not liked by the arthropod concerned. So it never used to go to that plant. But slowly, with the co-evolutionary relationship, these arthropods developed a strong reaction and they start going to that plant that is termed as karyomons. So we must exploit that principle in this. If we see that major route of action of insecticide, it is the three major routes: stomach poisons they can act, contact poisons, or fumigants. Remember that stomach poisons will act on the digestive system of the insect and then it will transmit the chemical to the other part of the body. Contact poisons act on the nervous system of the insects. So you must be very clear about the nervous system of the insect, and the last is fumigants act on the respiratory system. So it should be volatile, or it should be in gaseous form. We have to use it. Stomach poisons, neem leaves, seed and bark. These are few examples I have given. Contact poisons, pyrethrins and rotinones, fumigants, essential oils are available from spices. Then once you want to develop an insecticide, whether from chemical or whether from herbal, you must be very clear about the anatomy of the arthropod in question. It is its life cycle stages, because all these matters, their location and habits, habit of the adult insects, epidemiology of the insect concern. Because if you see anything, if you want to see anatomy, if you want to see cockroach, they come from the sewage, but they never get wet. So their body is covered with a layer, waxy layer or fatty layer. So if you are preparing a herbal chemical against it, so it should be an oil base, then only it will be applicable to their cuticle. Remember that. If it is on water base, it won't. Because when you see majority of the ticks, they are on the host, and rains are there, tick is there on the host, but that water doesn't give any impact to the tick. Even if you do in vitro trials, you wash the ticks first with the water, dry them, and then you apply the chemicals and you do the studies. So in that case, if you want to make a herbal insecticide or herbal acaricide, you have to be very clear. Body coat of the insect, you must be very clear, and its anatomy, its life cycle stages, their location. Suppose if we want to control ticks, and we are applying herbal chemical on the tick, on the host, but we are not trying to control its life cycle stages, which is lying in cow shed. One female, as you know, it gives more than 10,000 eggs. Again, the larva comes, and that is a major problem which we are facing in the field. Every now and then we are facing this problem that we have applied the chemicals, ticks have gone down, but within 15 days, again, the ticks have come up. So in that way, you should be very clear about the life cycle. Even in mosquitoes, you know, water bodies, unless you control the larva, you may not be able to control the adults. So habit of the adults insects is also important. What is their habit? You know, certain insects, particularly Tabanus, it has been observed that it takes a dip into the water after in the ponds or lakes. They take a dip regularly in hot summer. So we can use certain chemicals on the upper layer in the oil base. Once it takes a dip, it will die. Epidemiology of the insect concern, that is very important. See, remember that if you see the epidemiology, remember that cycloraphan insects, if you see, their life is very small. They have reduced mouth parts. Exceptions are there, which have teeth on the lower lip, and that is Stomoxys, Lyprosia. But rest of all the cycloraphan insects have a reduced mouth parts. So they exist in nature for very small period, hardly for 1 week to 1 month. And majority of the life cycle is in the host. It is the larval stages, whether it is hypoderma, whether it is gastrophilus, whether it is Oestrosovis. So you must be clear that 1 month epidemiology you should know in the area concerned where you are working, that when I should apply this chemical so that these flies do not infect my host, and you can even, a simple way you can control it. So once we are taking about the anatomy of insects, I have just taken the picture from the web only, but I just want to say there are few entries. You know, if you see the dorsal side of the insect, generally if you spray any chemical from the dorsal side, it never take penetration because it is thicker. From the ventral side, it is chitin layering is very poor. If you see, cuticle provides series of barriers. Some insects will have hairs, some will have wax, some will have threads like that, scales, etc. Generally, epicuticle is made up of fatty molecules on framework of 10 lipoprotein, as it has been shown. And these are the different ways they have been prepared. Nerve endings, which open in the last epidermis. They open in the epidermis, and lipoid sheaths of the nervous system with the initial entry through nerve ending at the cuticle, it happens. And these contact poisons also enter through mouth parts, labella, antenna, wings, ventral, cervical region, and thoracic sternite between the legs. Because from the ventral side, it is easy, because appendages are there, legs are there. That portion is a little bit, it is not so chitinous. It is a little flexible. From there, it takes the entry, and from that portion, the nerve is also very near to the near to attack on. The ventral side nerve cord is separated only by epidermis and basement membrane. Remember that Campaniform sensilla on trochanter is another vulnerable region. It is particularly for the cockroach. That's why Lakshman Rekha works very well, because from that also there is a this Campaniform sensilla opening. It goes to the nervous system and complete nervous system is destroyed. If we see the tarsi, then the tarsi of the leg, it is very thin cuticle it has, and it has chemoreceptors. And pulvuli do have gland openings. From there also these contact poisons take entry. Temperature also play a role in contact poison, because it is the oil waste, and in winter the entry is poor, whereas in summers entry is good. This is the picture for nervous system I have shown. From the ventral side you can see that all the nervous system is clearly visible in this picture. And from dorsal side it is difficult. From dorsal side, if it takes entry, generally it takes from the intersegmental glands. As you know, the head is made up of 6 segments. By the process, all the arthropods have come from soft annelid worms. The head was made up with the 6 segments, which 6 segments united together. The process was called cephalization. We cannot differentiate all the 6 segments. Only the 3rd segment was not having any appendage. All the 5 other segments of the head was provided with certain appendages, which were antenna, sympathetic, compound eye, and maxillae, mandibles. So like that, all the 6 appendages united together. So from their entry is very difficult, except it can go from the from the mouth parts. But if we see the thorax is made up of 3 segments, where the 3 segments join. From there the entry can take place very easily from the dorsal side. And in the abdomen also the segmentation is clear. In some insects we see 5, although it is made up of 10 to 12 segments, but it can be, in some it is visible are 8, some have united together, or the same way as it has happened in the head, cephalization has occurred. So in that way the complete joining of the segments have occurred. But thoracic, it is very clear, 3 appendages, 3 pairs of leg on the 3 particular segments of the thorax, wings, as you know, that's the basics. So when we talk about the anatomy of the insects, we know that stomach poisons are toxic if they are ingested through mouth, and are useful against the insect that have a biting and chewing mouth parts, such as caterpillars, beetles, and grasshoppers. Here we have major is beetles and certain stages, flea stages, which are coprophagous. So as veterinary is concerned, they are applied as sprays or dust in the animal sheds where bedding is there. So once these larval stages take this bedding, they will be destroyed. Remember that there are different enzymes are available in this digestive system of different insects. We are concerned only with here carnivorous insects, because they have proteases and lipases. If your herbal product is protein based, it will be digested. If it is a contact poison you are making, stomach poison you are preparing. If it is on the lipid base, it will be destroyed by the enzyme lipase there in the system. Regarding fumigants toxicity, large number of essential oil extracted from spices and herbs are being used. Even these tobacco leaves are also being used. Air enter through the insect body through valve. As you know, it is very clear, spiracle and pure whole tracheal system. Everybody is knowing here that what is tracheal system and how the air enter into the insect. It is the regulation of the body hemocoel and air sacs present in the body. Together they work together, so that air enters through the spiracles. So important thing is that action on eggs. When you prepare any herbal compound, because you have to kill all the stages. So eggs are enveloped by endochorion or exochorion. Insecticide must penetrate minute pores of the chorions or through micropores. All oil exert lethal effects by inhibiting hatching, by depressing respiration rate as they block the pores, as well as does not allow toxic metabolites to come out. Remember that we have to kill the eggs. If the eggs are oil resistant, then they can be destroyed with the phenol and creosoles, because you have to add these things, and they are not so harmful, phenols and creosoles. They soften and destroy the covering of the eggs, and then your compound will act. Certain chemicals harden the covering and blocks the gaseous exchange, and they kill the embryo, example lime sulfur, because they harden the shells, so a gas exchange is not possible. So regarding life cycle, I was talking about habits and epidemiology. Try to kill all the stages simultaneously. Important in ticks, as I have told you. Earlier I have said that if you are making any preparation for the adult, you have to make simultaneously control of the larval stages. Habit location whole group it is important. If you see nematocerian insects and brachycerian insects, all these nematocerian insect, all the 4 families which are of a veterinary importance, and brachycerian insect Tabanidae family, majority of them, their larval stages are near the water bodies. So you have to control that also when you are controlling the adults. Epidemiology helps you to use minimum insecticide for control of particular fly. Why I'm saying that? Because what we have observed that in Jammu, if this we talk about warble fly infestation in goat, we have seen, I have just shown a picture. You can see L3 in the body of the goat. These larvae generally laid in round about this months, July to August. If we apply even 5 microgram per kg body weight ivermectin, then when fly comes and lays its eggs, the larva comes out, it comes in this minute chemical, they are lost. So the flocks where we have placed this microdose of mini dose of boron preparation, there we could able to control this fly losses. Remember that these losses were immense. So that's why I'm saying when you see even Hypoderma lineatum, whether you see Gastrophilus, in Gastrophilus, what we have seen in horses of Katra, if you give medicines, chemicals to remove the L3 in early summer, then you are able to control the fly in the late part of the summer. So the very less flies are left over, so they lay less eggs on the legs of the horses, and the infection is reduced. Remember that we cannot eradicate them completely, but we can reduce their infestation. Then role of adjuvants in the insecticide, that is very important. Remember, we can use 1 or 2 adjuvants, we can use 3, but they should be able to working in together. We cannot use opposite qualities adjuvants. Adjuvants are designed to perform specific functions. They are solvent, emulsifiers, spreaders, stickers, buffers, thickness, synergist, and abrasive. Adjuvants also can reduce evaporation, foaming. Suppose we have using an adjuvant in our herbal insecticide and throwing it into the cattle shed, so evaporation should be slow, foaming should be less, spray drift should be good, and volatilization should be good. No, or less, it should be. No single adjuvant can perform all these functions. Remember that. So certain adjuvants even enhance the qualities of the compound which you are using. They boost the pesticide effectiveness or boost the insecticide effectiveness, and it also reduces the number of spray application. If we are able to control, if we add adjuvant, we can give 1 spray will be more good as compared to direct. If we supply that insecticide, we have to put 3 sprays. The spray adjuvant can be categorized into 2: activated adjuvants and special purpose or utility adjuvant. Generally, adjuvants are termed inactive, but certainly they are not, as they are protected through patents and proprietary regulation. They represent 90 to 95% of total volume of commercial formulations. Remember that. Remember that even what we were using earlier, we were using ash with chemical to play for dusting. Ash also blocks the pores of the eggs and doesn't allow the larval stages to come out, or the eggs are destroyed in with the simple ash. Also, people use ash still late in kitchen gardens. So I have here I have taken few common use plant as insecticide activity, and mode of action. Neem is very common, interfere with the and it has 3 types of qualities. Antifeedant, it interferes with the chemoreceptors, blocks sugar receptor cells, affects growth and molting, disrupt ecdysteroids. Ecdysteroids are used for molting, affect reproductions. It damage the juvenile hormone, prevent oogenesis and oviposition, and also interrupt mature sperm production. Commercial preparation, I have mentioned few of them, not all of them. Regarding Pongamia species or Crangen or Ponagomol, it too have antifeedant activities. It too has repellent larvicidal activities. Majority of people have used these activities against the larvae of mosquitoes. So commercially, one preparation I could get Dermizome. Mode of action, when we talk about nicotine, as you know, it is an autonomic blocking agent and acts like acetylcholine, but only at ganglia and neuromuscular junction, initially stimulating and then depressing them. It was banned in 2009 in European Union, as also been banned in USA. But leaves as smoke still used in many places to repel insects. Washing of animals with the water having fresh leaf is still being practiced in many parts of our country. Chrysanthemum, is one old one, as you know, pyrethroids and pyrethroids. What is the difference? Everyone is very clear about it. Pyrethroids attacks the nervous system of all insect and inhibit female mosquito from biting. In sublethal dose, they have insect repellent effect. They are harmful to fish, but are far less toxic to mammals and birds than many synthetic insecticides. That's why they are still in use. Synthetic pyrethroids. Many classes have come of synthetic pyrethroids, and they are still being used in the system. Then mode of action of Daris elliptica berm is rotinone. Here the root part was used, and the chemical produced was rotinone, which worked with interfering with the electron transport chain in mitochondria. It created a reactive oxygen species which damaged the DNA and other component of the mitochondria. It is few chemicals, few preparations which are commercially available. I have mentioned there. Then Cedrus deodora. It repels the insect, particularly mosquito and flies. It is the basic, it is in the, what do you say, Shivalik region or a higher region of the Himalaya, we find these trees, conifer. Basically, they are conifer. Remember that these conifer plants have juveline analogs. Remember that. It is available in the literature. And in Himachal, what I have observed, the spine trees, these pine trees which has a bifid needle, the oil, we call it as kale oil in local language. It is a variety of pinus only. And this cedrus or pinus tree with the bifid needles, that oil is available commercially in the market. Even in the villages it is available. It is the major component of Himax cream of Saharanpur Indian herbs. And it has all the three activities. Himax is widely used in our clinics, either Lorexins or Himax. They are used for as a repellents. They are healing properties, and even what is a three component it is being used. So it has that kale oil. Kale oil in villages of Himachal, generally they feed the animal. If ticks are there, they will directly give 50 ml of kale oil, and all the ticks fall down, all the lice fall down from the animal. This is age-old practice. So this oil has that property. So once I want to conclude here, if you want to go for herbal compound as insecticidal, you should first study the morphology of the insect, body coat of the insect, life cycle, which is the weak link where we can hit upon, and the habits of the insect. Another point is target system of the insect, which particular system you are going to target. Adjuvant, which adjuvant will be the best for that particular preparation. Identification of endophytes in plant used for preparing herbal ectoparasiticides. This is the important point which I want to give as a lead. Season of the year when plant material was collected, because content vary with the season. Altitude of the herb collection, variety, species of the herb, time of use of herb, post collection. Certain herbs, fresh, they give very good result, but once they are aged, they don't give the results. Why I'm saying that? Because with the time, with this varies. In Himachal clinics, what we used to observe, that all through the season we used to feed bamboo leaves to the animals, but 2 months, I think it was July, August, or some 2 months were there when we used to feed these leaves to the animals, ruminants. There used to be tremendous increase in the cases of volvulus and intussusception. So certain chemicals, alkaloids or season-wise alkaloids and other material increases, and these endophyte also varies, and they produce their factors. So in those 2 months, in Himachal, people don't feed bamboo leaves to the animals. So all the leaves have endophytes. All parts, I can say, all parts of the plant do have endophytes. It can be bacteria, it can be fungus, it can be any microorganism, and which along with the plant produce the things which may be of our use. All these factors changes the efficacy of herbal product, because as you know, herbal products, we do not get reproducible results. That is a major cause we are not able to register them. Endophyte act as a reservoir for just these are the statements which I am reading. If endophytes act as a reservoir for novel bioactive secondary metabolites such as alkaloids, phenolic acids, quinones, steroids, saponins, tannins, terpenins, that serves as a potential candidate for antimicrobial, anti-insect. Here I am worried about anti-insect. While plants sources are being extensively explored for new chemical entities, but nobody is looking towards the endophytes. They constitute a major source for drug discovery. Extraction of metabolites from endophytes is affected by various factors, such as season sample, which I have earlier discussed. There are quite big literature is available, and a large quantity of literature is available on metabolites. And Taxol anti-cancer product is a major which we have got through endophytes only. Huperzine, recently I have gone through from Huperzia serrata, can act as a cholinesterase inhibitor that can be exploited. When we see about juvenile hormones, I want to say that also a little bit. Juvenile hormones regulate many aspects of the insects physiology, development, reproduction, diapause, polyhensism. Polyhensism is, I mean to say, same genotype can produce many phenotype depending upon the climate change. And when juvenile hormones we talk about, it also impacts the eggs in the female insects. Remember that in development stage, as long as there is enough, from where it is produced, juvenile hormones is produced from the corpus allatum, endocrine gland just near the brain. And in the development stage, as long as their juvenile hormone, the ecdysone hormone is there, if these are equal, then larva to larva movement occurs. With lower amounts of juvenile hormone, ecdysone is more, then the larva pupates. And complete absence of juvenile hormones results in formation of adult. Synthetic analogs of juvenile hormones are used as insecticide, preventing the larva from developing into adult insects. Methoprin is one. Some juvenile hormone analogs have been found in coniferous. That is an important point which I want to say. So coniferous plants can be tried. Over 250 plants with active steroid analogs have been identified so far in plants, and it has been theorized that there are over 1,000 possible structures which might occur in nature. In last, I want to say, because they wanted, I should say few lines for the maggot debridgement. In the last, I want to say a few lines about the maggot debridgement therapy. As the organizer wanted to, these larvae which are being used as maggots being used for the treatment of the lesions. Remember that these maggots can be sterilized in the lab. It is Lucilia sericata's larva which we used, which gave which gave results. And remember that these maggots change the pH of these maggots. Hello, these maggots change the pH of the wound.
Sorry to interrupt. Dr. N. Jayakishan, Dr. N. Jayakishan, kindly unmute yourself. Otherwise, we will be forced to remove you. Dr. N. Jayakishan.
Hello. Over 250 active steroids. This I have completed. Maggot debridgement therapy, I have to say only 2-3 lines only. The important point of maggot debridgement therapy is that the larvae which we are taking, we can sterilize them, we can place them on the wounds which are which cannot be treated with antibiotics, or diabetic wounds. These larvae works very well. These larvae have 3-4 qualities. The first quality is that they eat the necrotic tissue, and it certain secretions it produce which have antibiotic qualities, and it kill that particular bacteria in the wound. And the bacteria which it is not able to kill, it digests that, it orally take that bacteria into its gut and destroys it, and changes the pH of the wound, which help in healing. It also produce certain growth factor when larva mold from L1 to L2, L2 to L3. Remember that we keep larva only for 24 hours. So in that small period only the release excretion of secretions, growth factors are released. Those growth factors are conserved in host also and for larva also. So those growth factors works for our wound also. So our tissue give very good healing in that cases. And they produce minute current because of moving into the system. That also help in healing. With that, I want to end my talk. Thank you, everybody. Any questions you can ask me. कैमरे पे कैसे होगा कैमरा
ये वाला वो कर लो ये वो
Sharing the sir.
Chrome.
Yeah.
Sir, thank you very much for your insightful presentation, sir. It's a teacher like you, sir, who freely gave up their own spare time to ensure that the next generation is better informed and educated.
Thank you. Any questions?
Yes, sir. There are a few questions from the participant side, sir.
Okay.
The first question is there any product of insect growth regulator commercially available in the market?
Yeah, it is available. If you go into the net, you will get that. And here I want to say, we can see, in addition to that, we should not apply insecticide directly on the insect. We should use certain attractants in the cow shed. We should place as they are using in the agriculture side. They are not applying nowadays chemicals onto the crop. Okay. They are applying right on the wall. You can place an envelope where you can apply attractants and contact poison to it. As the insect come to the attractants, it falls into that bag and you collect the arthropod. So you need not to apply on this. Growth regulators, these are available in the market. You can go, there are a lot of review are there. All insect growth regulators are available. IGRs are available.
Okay, sir.
In India, we don't have much of the availability.
Okay, sir. The next question is why there is no report of resistance against herbal acaricides?
Because everybody is using herbs of his area or specific area only. And herbal compounds are not giving reproducible results. I'm telling you that I told you. It depends upon the herb collected from the area, altitude, endophytes in that. Nobody evaluates the endophytes in them. Because endophytes, fungal endophytes, many chemicals have come from endophytes, remember that. Our many antibiotics. So if we are not able to say same plant may not give you the result if it is of higher altitude, but of the tropical side, it may give results. So it depends upon the endophytes in them. So we don't get reproducible results in herbal compounds. That is a problem in registration.
Okay, sir. The next question is, sir, if somebody successfully isolates an effective molecule from a traditionally used plant, can it be patented?
Yeah, it can be patented if it is working well. But generally, herbal compounds doesn't work in single biomolecule. Doesn't work. Although we have nicotine is there, azatectin is there, from rotinone is there, from Daris, they do work. But their stability has to be produced. Why we do these adjuvants? Adjuvants we give stability to these chemicals in the nature.
Okay, sir. Sir, next question is, housefly is a big problem in cattle shed.
Yeah.
Okay, sir. So is there any alternative measures? Sir,
Yes.
This is a big problem in Haryana also, in particular in poultry farms.
Yeah, my opinion is that we should use the juvenile hormones. If the poultry farm is just like on the net and feces is collected beneath it, generally what type of poultry farm I am talking, because net is there, poultry are there, and fecal material is collected just beneath it. In that cases, we can spray these juvenile hormones. So the larva will not molt. It will remain as larva only, and slowly it will destroy.
So, sir, the next question related to this is juvenile hormone, sir. Is there any commercially available product based on insect growth regulator or juvenile hormones, sir, in the market?
Juvenile hormones, because I am not clear about it. It is available or not. That I cannot. Frankly speaking, I have not gone through it. But once they remove that particular gland, endocrine gland from the insect, the studies have been made. So then the larva molted to the next stage because of ecdysone hormone, but it did not turn into nymph, and nymph was destroyed. That studies have been made.
Okay, sir. The next question from Dr. C.P. Sornagar, sir. Sir, as we know, the inherent properties of plants are mainly by secondary metabolites. Yes, and these secondary metabolites are mostly produced by the plants during stress condition.
Yeah.
So, can we get the same extent of secondary metabolites in garden-grown plants?
Yeah, if stress is there, because that only I'm saying, not even stress, the protection to plant in stress condition is given by these only endophytes. They provide those biomolecules to overcome the stress, because endophyte is a very big subject. If you read that, that only provides the to withstand the stress in plants also.
So last two questions. Sir, is there any foreign formulation available on ivermectin for control of verbal plaques in sheep and goats?
Yeah, we have. I don't remember the name. Dr. Anish can give you because he is using it because it is his project.
Okay, sir.
We have commercially available. I don't remember now in my. It's not in my mind, but it is commercially available. And Dr.
Sir,
Anish is using.
Okay, sir. The last question, sir.
Yeah.
Sir, what strategy will be adopted to treat tick infestations in free-ranging wild animals?
In wild animals.
Yes, sir. Wild animals.
It's difficult, it is difficult in wild animals because wild animals, they roam everywhere and we do not know where the larval stages are existing. So we have to go certain nematodes they are trying in the world, certain nematodes which are eating the larval stages of the ticks. Well, the studies are going in the USA. I think Dr. Nirbe has worked on it. Certain nematode stages are there which are working on the larval stages of the ticks which are in the nature. So they are trying those larvae.
Okay, sir. Thank you very much for such a wonderful interactive session, sir.
Thank
Now
you.
I request Dr. Snail to propose vote of thanks. Dr. Snail, please.
A very good morning to everyone.
Good morning.
A wise man said, an ignited mind is the most powerful weapon on the earth, above the earth and under the earth. And this ignition is only got through such kind of deliberation. Sir, you have touched a very critical area because we all are facing the ectoparasitic problem. I, Dr. Snehil Gupta, would like to take the honor to express our gratitude to our Dr. Rajesh Katoj, who wholeheartedly accepted our invitation for the webinar series. Herbal ectoparasiticides are the need of the hour. It was indeed a delightful presentation, sir.
Thank you.
Such a compilation on the herbal ectoparasiticides is rare to find anywhere else. Your exclusive work on the maggot debridement therapy will indeed open new avenues in the veterinary parasitology. Sir, your message was exactly what we need to hear. Thank you very much once again, sir, for sharing your lead with the budding parasitology.
Thank you. Welcome. Welcome.
Thank you. Now over to Dr. Maran.
So, on behalf of the organizing committee, I feel privileged and honored to introduce our next eminent speaker, Dr. Rajat sir, Principal Scientist, ICAR, IVR. He has done his Bachelor and Master from CCS and Doctorate from G.B. Pantnagar University, Pantnagar. He has handled 6 research projects as Principal Investigator and 9 co-principal investigator. He has published 138 publications, 9 books, 16 book chapters, and he has developed 3 mobile apps. He has guided 6 MBSc and 4 PhD students as major advisor. He also acts as course coordinator in 21 days ICR-sponsored winter school on molecular biological approaches for diagnosis and control parasites organized in the Division of Parasitology, IVR Nagar from 2nd to 22nd December 2013. Course coordinator he has also acts as course coordinator for 1-week training program for veterinary officers of Department of Animal Husbandry, Uttarakhand, on the topic laboratory techniques for the diagnosis of common parasite diseases of domestic animal, organized by Division of Parasitology, IVRI, during the year 2012. He has also bagged many national and international awards, as well as he is the recipient of so many awards. Sorry for the interruption. So he has bagged CSIR Senior Research Fellowship in Veterinary Parasitology for the year 1997, LP Banerjee Oration Award 2017 by IABP for distinguished contribution in the field of veterinary protozoology. Dr. Kalyan Sundaram Award in the year 2018 by IABP for distinguish control in the field of poultry parasites. He has also bagged 3 times Best Article Award. He is Alvar Best Article Award for the year 2015, Best Article Award for the year 2007, Best Article Award in the year 2011 by Indian Association for Advancement of Veterinary Parasitology. He has also got Best Poster Award 3 times in 2004, 2017. He attended many conferences, both international and national conferences. Now I request Dr. Rajat sir to please go ahead with his speech. Over to Dr. Rajat sir.
Thank you. Thank you very much. I welcome you all to today's presentation. And are you able to see my screen? All of you? Hello?
Yes, sir. Yes, sir.
Okay, okay. Thank
Kindly
you
make
very
it full
much.
screen, sir. Kindly
Yeah?
make it full screen, sir.
Okay, yes.
Full
I
screen,
will.
sir.
Yes, yes, I am doing it. Okay, is it okay?
Yes, sir.
Okay, at the very onset, I would like to thank the Dean, College of Veterinary Sciences, Hisar, Dr. Divakar Sharma, and Dr. S.K. Gupta, Dr. Vora for inviting me to present today's seminar. I would also like to thank the members of the organizing committee for providing me this platform to present the seminar. My topic for today's seminar is the control of chicken coccidiosis, and I will be discussing about few of the challenges and the opportunities and the changing trends in the disease. Now, before going into the details of the control of coccidiosis, I would like to highlight certain points of the Eimeria, the genus Eimeria. Now, the genus Eimeria, it belongs to the phylum Apicomplexa, and they are single-celled protozoa. Now, the characteristic feature of this genus or this phylum Apicomplexa is the presence of the apical complex, which mainly comprises of 5 important organelles, I will say: polar ring, conoid, rostries, micronemes, and dense granules. Now, these 5 organelles, they are directly related to the penetration as well as the movement of the parasite within the host cell. Now, without the secretions and excretions by these organelles and without the movement or the wave that is produced in the microtubules, the parasite will not be able to move or enter into the cell. So the major focus of today's presentation will be on the use of these organelles for control of coccidiosis, which will be I will be dealing in the later stages. Now, the important thing is that the life cycle of coccidia is self-limiting, but it is very rapid and acute form of life cycle. Self-limiting only when in absence of reinfections. If there is no reinfection in the animal, the organism will complete its life cycle and will pass out in the poultry droppings. Another important thing is that is utilized or that is helpful in actually control of parasite is that the parasites do not replicate productively outside the host. I am talking of the sexual reproduction. It does not occur outside the host, and all of you know that it causes a very important disease, that is the chicken coccidiosis. Now, in fact, there are 7 Eimeria species that are well recognized around the world: Eimeria cerulina, bruneti, maxima, mitis, nicatrix, precox, and tenella. Tenella being the most pathogenic, and the 3 most important are those causing hemorrhagic coccidiosis are put in red. That is Eimeria bruneti, nicatrix, and tenella. All these 3 cause hemorrhagic coccidiosis, while the others they cause non-hemorrhagic or they cause the intestinal coccidiosis in birds, basically. Now, beside these 7 species, all of you must be knowing there are 2 more species that have been given in the books or that have been debated by a large number of people throughout the world. One is Eimeria hagani and another one is Eimeria mewati. Now, for your information, Eimeria hagani has only been once reported, and that was by Levine when it was found. After that, as far as my knowledge goes, Eimeria hagani has never been reported after that. Eimeria mivati, they say it is a, either a, it is, you can say it is a subtype or a variety of Eimeria acervulina. Now, these Eimeria species, they are global in distribution. All the continents, all the places where chicken are grown throughout the world, you will find Eimeria infection in those. And it infects almost all the flocks around the world at one time or the other. Though with the advent of the cage system, the incidence might have gone a little bit down, but the parasite is still present in each and every flock. So a very important characteristic feature of the genus Eimeria is the oocysts are very tough. They can resist adverse environmental conditions for quite some time. They have high reproduction rates. The zygogony, there may be multiple generations of zygogony resulting in production of large number of merozoites, and this will lead into formation of large number of macrogametocytes and ultimately a large number of oocysts. Another very peculiar feature of this different species is that there are 7 recognized species, and each species is capable of causing a different kind of a disease in the animal. Though the mixed infections are almost a rule, very rarely you will find a single species infection. The parasite has got is highly immunogenic, with Eimeria maxima being the most immunogenic species, and the species immunity is also specific, means that the birds immunized with Eimeria tenella are protected against Eimeria tenella, but not against any of the other species. Though cross protection between the species have been reported now of late. Now, there are few things I would like to point out regarding the life cycle, that they are highly host specific. The species of chicken will infect only chicken. They will not infect any other host species, not even other kind of bird species like turkeys or other leave-aside animals. Only single host is involved. They have a direct life cycle. They involve only a single host. No intermediate host is required. And I have already told you the life cycle is self-limiting. The multiplication rates are very high, and a large number of infective stages build up in the environment. And that's why control of at this stage, that is the environmental contamination, at this stage is very important in case of coccidiosis. You can use any medicine, any vaccine, but if you are allowing the buildup of infection in the environment, you will not be able to control coccidiosis at any cost. Now, regarding the situation in India, it is one of the top 3 economically important diseases of poultry industry, I will say, and with the annual economic loss of more than 1.2 billion rupees. And that loss is only estimated in terms of the cost of morbidity, mortality, and production losses. Add to this the cost of your anticoccidials used in the market. This will be at least 3-4 times of this. And almost all the chicken in India, 80 million broilers and more than 410 million layers that are produced in India are at risk to this disease. Now, a very extensive survey was carried out in India under SIDLIT project, which was an anticoccidial vaccine development in almost 9 states of India, 4 North Indian states and 4 South Indian states, and 1 Western state, which is actually the poultry producing belt of India. Mainly it is Andhra Pradesh, Karnataka, Tamil Nadu in the south. Western it will be in Maharashtra, and in north it starts from UP, Punjab, Haryana. So this is the poultry producing belt of India. In almost all these states, approximately 500 farms were covered in all these states. Half of the north part was covered by IVRI, whereas the south part, it was covered by LUVAS. And it was found that almost 80% of farms were positive for coccidial infections at a given time, at the time of sampling, I will say. And further studies revealed that Eimeria tenella was the most common species throughout the country, followed by Eimeria mitis, Eimeria acervulina, and Eimeria nicatrix. Then further studies were carried out in that project to study whether these are the only species that are found in India. And it was found that besides the various species of Eimeria prevalent in India, when genotyping was done using ITS markers, it was found there are actually three operational taxonomic units of Eimeria that have been recognized globally. And out of these three, they are known as OTUX, OTUY, and OTUZ. Out of those three, two were existing. And a very important thing that was noted was that if you see the global trends, these operational taxonomic units, they are only present in the forms that are located below 30° north latitude. Means they are mainly located in the southern hemisphere of the globe and not in the northern hemisphere, and they assume great importance in the control of coccidiosis. Now, before going into details, the most important, a very important factor for designing a control strategy for any disease is to know its prevalence, and another important thing is how to diagnose it. Now, you know, I discussed there are 7 species. Diagnosis of coccidiosis, whether the bird is infected with coccidia or not, is not that difficult. But which species of Eimeria is involved in the disease, that assumes a great importance. Now, you can see the first line in the picture, where you will find that the red portion shows the part of the intestine that is infected by different Eimeria species. And the second and third rows, you see the size of the oocyst. Now, if you see the picture, sometimes it will be difficult. If it is a cerulina, then how will you differentiate it from peacocks? Both of them infect the anterior part of the intestine. If it is maxima, how will you differentiate it from mitis infection? And sometimes Bruneti and Tenella can also be confused. And a mixed infection of different species, in those cases, how will you diagnose which species is actually prevalent and which is the predominant species in the form that is causing the disease? And you may think of that I will examine the fecal sample and look for the oocysts which are present. And again, you see the size and the shape of the oocysts. Of course, Eimeria maxima can be identified because the oocysts are the biggest. But what about Eimeria bruneti and Eimeria tenella? If you see the oocysts, they are almost similar in shape and size as seen in this photograph. Similarly, it is sometimes difficult to differentiate precox, nicatrix from the size of the oocysts. The mitis oocysts are comparatively easier because they are the smallest in size. And cerulina, once you gain experience, you can also identify Eimeria cerulina based on the oocysts because they are slightly cylindrical as compared to others. Now, to identify these species at the field level, it is a very innovative, you can say, computer-based technique was launched by Carvalho in the year, I think, 2008 or 9. And the software that he developed was Coccimoth software. And this software, it identifies the Eimerian oocysts of chicken or rabbits only, and that is based on the shape and the size as well as the curvature of the oocysts, the angles of the various porosis that are present in it, and various other morphological features it has taken into account. And the photographs are taken and they are loaded on the software, and based on the morphology of the oocysts are identified. And the results will be like the oocyst that you have uploaded is almost 90% similar to Eimeria tenella and maybe 10% similar to other species or whatever it is. So you can identify it on this basis then, but the accuracy is not that very high. It is around 70% accurate. Other things that can be used are the SCAR multiplex PCR assays, and that we use in our project, in the SIDLIT project, for identification of a number of species. And in this, it is a very user-friendly method. All the 7 species can be amplified within a single tube. But again, since it is a multiplex PCR, the sensitivity of the test is comparatively lower than the nested PCR assay. That is the most frequently used technique for the identification of, sorry please, for identification of Eimeria species of chicken, and it is based on ITS1 regions of the Eimeria genome. And based on the nested PCR, we identified the different species of Eimeria throughout the country. And as I told you that we covered 9 states in the country, right from UP, Uttarakhand, Punjab, Haryana, Madhya Pradesh, and Andhra, Kerala, Pondicherry, and Karnataka. And you can see that Eimeria tenella, which is the yellowish one, is the most prevalent species in the country. In almost all kinds of flocks, it may be a broiler or a broiler breeder, or it is a layer or a backyard poultry. In all the cases, in all the states, it was Eimeria tenella, which was the most common species. Leave aside the age group of animals, whatever it is, but it was the most common. Whereas Eimeria precox, it was only recorded in the northern parts of India. Southern India, we could not find Eimeria precox infection. Third important thing that was recorded in the study was that Eimeria nicatrix infection, it was very common in the north as compared to the South India. So the two most pathogenic species, Eimeria nicatrix in the adults, as well as Eimeria tenella, which is more important in the broiler industry, Eimeria nicatrix more important in the layer industry. Both were very highly prevalent in the North India as compared to the South. All broiler production systems were having all these Eimeria species as well. Now, with this background, once we know the species composition, whatever it is present, we come to the main topic, that is the control of chicken coccidiosis. Now, like any other disease, coccidiosis can also be controlled by various ways. And the most important is, I will say, besides chemotherapy, is the managemental practices. And I put managemental practices on the first in this regard. Managemental practices are very important, followed by chemotherapy. Then nowadays, immunoprophylaxis is slowly coming up in India as well. Some vaccines have come up. Then botanical and herbal control. Market is full of herbal anticoccidials throughout the globe. Now, very feed additives, particularly probiotics, are coming up for coccidia, and genetic resistance of the breed is also being studied in some of the breeds in various parts of the country. Now, starting with the managemental control, and the first thing in the managemental control that I will discuss is the biosecurity measures for control of coccidiosis. Now, the main biosecurity, the purpose of biosecurity is to reduce the contamination among the birds within a single flock, and also reduce cross-contamination between the flocks. So both these things are very important. And the first and the foremost thing is that we should separate the forms from each other by proper fencing. The fencing is the most important thing, because it will also help in preventing the entry of birds, animals, and even rodents and pets between the farms, so that infection present in one farm or in one shed is not carried to the next farm or the next shed. That is one thing which is very important. Now, you might have visited a number of poultry farms, and some of you, not all, but when you enter a poultry farm, there is generally a placard there. It is placed: visitors, please follow the biosecurity, farm biosecurity. Please contact the owner of the farm before entering. Do not enter without permission, whatever it is. And even if you have contacted, even sometimes when we used to go to poultry farms, we face the challenge of getting permissions from the owner. If owner, which is very, who is educated and he knows the concept of biosecurity, will not allow you to enter the poultry farm very easily. So the entry of persons, outside visitors to the farms, it should be restricted because we are a very common source of infection. Even the veterinarians and other visitors, we can carry the infection from one farm to another or from one shed to another through our shoes, through our clothing, or through our hands that are not properly washed. So we should change our boots and clothing while entering into the farms and handle the birds only after strict sanitization of your hand. A sanitizer should always be placed due to COVID-19 situation. Now everyone is using hand sanitizers and it is very helpful. Then another important thing is the movement of vehicles between the farms should also be restricted because vehicles are also, they also act as carriers, as physical carriers of the infection from one farm to another. And similarly, poultry equipment, if they are at all to be used between two sheds or between two farms, they should be sterilized first, or they should be properly sanitized first before transferring them to the next shed or before using them. So biosecurity plays a very important role. Then the second important thing is the environmental, under managemental practices is the environmental management. And this environmental management includes good ventilation. Proper ventilation in the poultry house will help to remove the moisture or will keep the litter dry. So it will help in keeping the birds healthy as well. So we should have a proper ventilation. If you do not have proper windows or other things, then try to put on exhaust fans and air conditioners if possible in your poultry farm. Then remove the sources of water besides feeder and waterer. You should not allow any other source of water inside the shed at least. Keep the litter dry by frequently turning off the litter if it is a deep litter system. In cage system, litter is not a much big problem, but the cages in cage system, the cages should be kept at a little bit height so that the lower rack of cages does not come in the contact of the feces of the birds. And because if they will come in contact with the feces of the birds, at least the birds that are present in the lower rows of the cages, they will take up the oozes from the environment. Then keep the water and feeder at a height which is at a level with the back of the bird, because this will help in preventing the birds defecating into the waterers and feeder. And if they are carriers of the infection, then they will not it will be an easy way to spread the infection if they defecate into waterers and feeder. Then another important thing is to maintain optimum bird density. Now the bird density, how much bird density, it varies. But what is ideal is that it should be around 1 square feet for the broilers and around 2 square feet for the layers. So if you manage it with this ratio, you may not be at a very very good, you can say, you may not get very good returns. So the farmers, they vary it. Then do not allow the birds to be under stress and provide them good and feed and water. You all know about this. And another important thing is that leave the shed empty for at least 14 days between the two flocks. This is also very important. Once you are replacing a flock, you should leave it without birds for at least 14 days. And during this period, you can clean up the flock, dry up the litter, stack it, and what all those precautions can be taken up during this period. This will help in removal of the oocyst and build up the oocyst in the poultry farm. Then keep the older birds away from
గవర్నమెంట్ నా దగ్గర ఉన్నది బాబు. నేను ఫస్ట్ టైం ఇక్కడ హెల్ప్ లైన్ ఫోన్ చేయలేదు. VISIT ఆల్రెడీ.
Okay, so this completes. This was a little bit about the managemental practices. Now the major mainstay of the control of coccidiosis in chicken is by chemotherapy, or you can say chemoprophylaxis. It is a major control of prophylaxis is use of various drugs, and drugs are very specific. They are not used for even generally related parasites, other epicomplexans, like even they cannot be used for plasmodium. They are highly specific, meant for coccidiosis. But the important thing is they have a residual effect. So the drugs should be withdrawn at least few days before the slaughter. The rate varies. It varies from, there are drugs like salinomycin, if I am not wrong, it requires only 1 day of withdrawal period, whereas a number of anticoccidials like sulfa and others, they require at least 7 day of withdrawal period. So that is a very important thing. The drugs against coccidiosis, they started coming up in the, sorry, just a minute. The drugs, they started coming in the market in around 1945. And after that, a number of drugs, they came up, and till inophores, they were launched, and slowly and slowly, though I know after inophores, no new class of drugs, they have come up in the market. The problem is that once the drugs were introduced into the market, drug resistance also came up along with it. And there are drugs in which resistance appeared within 1 year of their coming up in the market, and there are drugs which which resistance appeared around 6 to 7 years after coming off the market. And if you are not properly using them, or you are not strategically using the drugs, then cross resistance may also occur. So these are few of the common anticoccidial drugs that are available or that are used globally. Red ones are the groups of anticoccidials, and those given in the brackets, they are the compounds that are available. Now, if you see the global coccidial market, it is in last year it was around 331 million US dollars, and it is bound to increase 1.5 times in another 5 to 6 years, and in another 10 years this market will almost double. So this is how the market is increasing day by day. The anticoccidials, those are the only viable things that are available throughout the world, and that's why the market, and if you see the share of Asia Pacific region, the green part, it is the maximum, which is followed by the American subcontinent, particularly the North America. Now, if you have a very good drug, a very perfect anticoccidial, and you are following great management, then it will give a perfect balance. There will be no infection in the flock. If there will be no infection, there will be no immunity in the flocks, but no such effective drug is available nowadays because of presence of drug resistance. The drug resistance has totally changed the scenario. It has the infection rates have increased, but the plus point is with the infection, the immunity also comes into play. So immunity also increased, and also the outbreak numbers have increased. Why? Because if you are using these drugs in a non-synchronous way, then the infections will also be non-synchronous. There will be buildup of infection in the flock, and disease will occur. Now, I was talking of the introduction of anticoccidials and the resistance that came up in the market. The first anticoccidial that was introduced in the market was sulfaquinoxaline. It was introduced in the year 1948. Along with a few days after it, nitrofurazone was also introduced. But you see, within 6 years, the resistance against sulfaquinoxaline and nitrofurazone was reported from the USA. And you see the last two drugs, lactaloside and buconolate. In both these cases, the resistance appeared within 1 year of introduction of the drug. and of late, no new drugs have come up in the market. Now, if you see the Indian scenario, you can see the list. Almost all drugs, they have anticoccidial resistance has been reported. And if you see the references that I have quoted, most of the references, they are from Hisar or from LUVAS, the work that is being carried out at HAU Hisar. Most of the workers in Haryana, as well as in Punjab, or Swell Punjab also. So Punjab and Haryana, they have reported more resistance against most of the anticoccidials. Though farmers, the poultry owners, they report from throughout the country resistance has been reported against these drugs. Now, what are the consequences of this drug resistance? There will be poor weight gain in the farm. There will be increased contamination with the oozes. Severe parasite-induced lesions will be seen as a result of the disease. Mortalities will increase, and as such, we require some ways to manage this drug resistance. How to combat this drug resistance? So the first thing is, whenever you want to manage any kind of thing, is you should know the status of the disease, as well as in this case, as I am talking of resistance, so you should know the status of drug resistance. Unfortunately, we don't have any in vitro methods till date. So only thing is you have to go for an in vivo testing, and various indices, that is anticoccidial sensitivity tests, have to be done. And for this, two most commonly used indices are the global index and the anticoccidial index that are used. And these are based on the weight gains, oocyst reductions, lesion scores, as well as the feed conversion efficiencies in the bird. Then we also need to have some mathematical modelings can be done, but so that we can understand the dynamics and the drug resistance status in various production systems. However, monitoring drug resistance is not very frequently done in India, and we should focus on it. Then how to combat this? The important thing is that we should introduce either shuttle programs or the rotational program. Now, in rotation programs, drugs are rotated from one batch to another batch. So within a single batch, we will use a single kind of a drug throughout its life. But once we are changing the batch, then another drug from another class of anticoccidial is used. So this is one way of controlling resistance. Then the second one is the shuttle programs. In this, two or more drugs belonging to different classes, they are used as anticoccidials within a single batch. So what we do is that we use one anticoccidial in the starter and another one in the grower ration, and then we return back to the first drug in the finisher ration. So this is a shuttle program. Then another form of program are the bio-shuttle programs, where we use a combination of anticoccidial vaccine and drugs at different ages during a batch that is grown. So for example, on day one, a vaccine will be administered, and around 14 to 28 days after this, an anticoccidial drug will be given, from one group, and then in the finisher ration, another anticoccidial from another group will be given on 28 to 38 days. This is generally practiced in case of what is practiced in case of broilers. Then another thing is we should always try to restore the sensitivity by introducing drug-sensitive coccidia into the flock. So one thing is you can use drug-sensitive laboratory maintained lines. You can frequently infect your flock with a very low doses, maybe 30 to 50 uses per bird in the farm with the laboratory maintained line. Or you can use a mixture of a sensitive or resistant strain and then propagate them in unmedicated chicken in the flock. This will help in what the advantage will be, that it will help in development of a very strong immunity in the flock. And the third one is you can use non-attenuated live coccidial vaccines in the flock, because these non-attenuated live vaccines, they are generally made from drug sensitive strains of these ooses. So generally what is being done when you are using these kind of, you want to restore the sensitivity. In the picture I have shown, this picture I took it from a program that was developed by, I think, one pharmaceutical company of US. They say that within a year you can have at least 6 or 7, 6 batches of chicken. So what they recommended is that in the first 2 batches, or in the first batch, you go for the use of coccidiosis. In the subsequent 2 or 3 batches, use a vaccine. And lastly, in the last 2 batches, you again shift to the coccidiosis. So this will help in diluting the effect of anticoccidial resistance within the flock. Now, another way of combating the resistance or for controlling coccidiosis as a whole is the use of anticoccidial vaccines. And various vaccines are available. The most common ones are the live vaccines. There are 2 types of live vaccines. One are the virulent, those developed from virulent strains of the parasite, and second are those which are attenuated vaccines. So both these are commonly available in different parts of the country. They are increasingly being used, particularly in the broiler breeders and the layer birds. I have already discussed, they introduce drug sensitive strains in the flock, and there are several of them in the market. The important ones were being Coccivac B, Immucox, then Nobilis Cox-ATM, Advent Coccidiosis vaccine. So there are different vaccines, live vaccines, that are available in the market. None of them is being used, I think, in India. Live virulent strains, they are not being imported into in our country, and no live virulent strain vaccines are available in India, as far as my knowledge is. The use of these vaccines, it requires a very strict monitoring, because we are using a very low dose of ooses in these. Hardly 30 to 50 ooses are injected are orally administered in the bird. 1 ml vaccine contains around 30 to 50,000 ooses, and they are 1 ml is diluted in around in 1 liter of water, and this 1 liter of water is then fed to 1,000 birds. So 30 to 50 ooses per bird will come, and this will cycle into the bird. Ooses will be passed out in the feces. Again, these will be consumed by the birds through oral route. Again, and in such a way, a kind of strong immunity will develop in the bird. But we should follow, again, some biosecurity during this period and management. That is, you should keep on turning the litter and other things, keeping them dry. Otherwise, infection will build up, and it may also result in the clinical disease in the bird. Now, the safest vaccines that have been produced till date are the vaccines that utilizes the precocious strains of the parasite. That is, we attenuate the parasites and use them in a vaccine. In the precocious strains, what is being done is that we collect the earliest produced ooses. That is, the ooses that are first produced at around between 4th or around early 5th day. Only those ooses are collected, and they are then passaged into next group. They are sporulated, and then they are passaged into the birds. Next group of birds, again, earliest ooses are collected, and this is continued for at least 20 to 30 passages. A time will come that the parasite will lose its pathogenicity, but it will retain its immunogenicity. So that is the advantage of these precocious strains. The important thing is that these precocious strains, they are genetically stable. We do not find much of genetic variability in these strains once a precocious strain is established. And we can develop precocious strains against all Eimeriia species. They are fully attenuated, but they are attenuated but fully immunogenic. However, they are expensive and require a lot of chicken for propagation. So that is a problem with this. They are very commonly used in the world, and over the years, last 4-5 years, few of them are also available in our country. I think Livacox is available in India. Hipra is also Hipracox. Is also coming up in India. Hue Pharma, they brought up Advent in India, which also has this precocious strain of Eimeriia. So some of them are coming up in India. The third type of vaccines are the subunit vaccines. These are the stage-specific vaccines which have been derived from various life cycle stages of the parasite. And the parasites, they have been identified on the basis, the antigens, they have been identified on the basis of the host parasite host parasite interactions, their invasion, and the replication. They are based on these. They have been identified. Then another way of identifying these antigens are on the basis of genome, transcriptomes, and proteomes. And these resources are now available, and they have been used for designing various subunit vaccine candidates. The genome of Eimeriia is quite large. Eimeriia tenella genome, I think, is around 5.5 billion kilobases. Based on this, only one commercial vaccine is available, that is Coxabic, which utilizes Eimeriia maxima gametoside-derived native antigen. It is a native antigen vaccine which is available. Besides this, various recombinant proteins are being tried. DNA vaccines are also a good alternative. Now, where are we stuck? Different live vaccines, I have told, they are there. Attenuated vaccines are there. Subunit vaccines are also coming on. But they all have certain drawbacks, which I have listed here. I'm leaving this slide. Now, the thing comes how to identify which vaccine candidates we should use. And here, I told you in the first slide, the role of apical complex. The organelles that are involved in locomotion as well as in the penetration and establishment of the parasite. The role of these organelles, the apical complex, is here. That is the penetration and the establishment of the parasite within the host. So we have surface antigens on the surface, microneme proteins are there, apical membrane proteins are there, rough-free proteins are there. All these proteins, and a number of other proteins are there which help in the penetration and establishment of the parasite within the host. And here I have listed a few of the antigens that have been globally tried. There may be few more, but these are the most common ones that have been tried, like surface antigens, immune map protein, they have been tried of late, ROPS and RONS, that is ROP3 associated proteins, microneme proteins, a number of microneme proteins are there. Again, another microneme protein which is very commonly, which is a very good vaccine candidate is apical membrane antigen 1. Then profilins, they have been tried. Refractile body proteins have been tried. Vaccines have been tried from other like GADPH and other these metabolic proteins have been tried. And lastly, in this list comes macrogamete proteins, those derived from the oocyst that are important in oocyst wall formation and maybe transmission blocking vaccines like GAM56, GAM86, GAM230, and GAM20. So these candidates have been tried. Now the important thing is the delivery of these protective antigens, how to deliver them. People have tried various common ways in which they can be delivered. Native proteins, I have already told, they have been tried, and the only commercial vaccine that is available is on the based on the native protein. Then you can express these proteins in various expression systems, eukaryotic expression system, yeast. Nowadays, transgenic work is going on in different parts of the world, I will say, on developing transgenic Imeria vaccines. Means they are incorporating Imeria maxima, you can say Imeria maxima apical membrane antigen genes into Imeria tenella oocyst. So what will happen, or the other way around, or even into Imeria metis. So what will happen is that if you feed the birds with these strains of Imeria tenella or Imeria maxima or Imeria metis that are carrying the oocysts of or that are carrying the proteins, immunogenic proteins of other Imeria species, then once they are introduced into the host, they will multiply, they will release those proteins, and they will help in cross protection, I will say. Then various adjuvants, they can also be used along with these proteins. You can number of adjuvants, cytokine, even cytokines themselves, interferon gamma and interleukin 2, they have been used as adjuvants. Other adjuvants, water in oil adjuvants, they can also help in enhancement of immunity. Now, globally, if you see that it is the DNA vaccines and the recombinant proteins either expressed in eukaryotic expression system or in yeast or used in DNA combination, they have been used. And I have here copied this list from one of the reviews from, I think it was from Dammer Blake. So this review, from that review, I copied this table. And here you can see that immune map protein showed very good protection, and along with this profilin 3 rhomboid like protein, it gave 76% protection, microneme protein 70 up to 79% protection. So the similar kind different proteins, they have produced different levels of protection in birds. I am talking this protection, what I have shown is here is in terms of reduction in oocyst count. Now, some work we also did in IVRI. We tried to explore various proteins and various types of antigens for developing some immunogens for control of coccidiosis. We use live Imeria tenella, sporulated oocyst. We tried with sporulated oocyst antigen. We tried with sporozoite antigens. We also used various recombinant proteins for these vaccine trials. So what we found that if you we divided it into two parts, if you say the best are the sporulated oocyst given themselves. So here what we did, we fed the birds with 1,000 oocysts of Imeria tenella on 7 and 21 days of age, and we could see that it resulted in around 74% oocyst reduction as compared to control. Whereas when we used the antigen from the same oocyst, it resulted in around 72% oocyst reduction. And if we use the sporozoite antigens derived from this, it resulted in around 57 to 58% oocyst reduction. So it means that it is not like that the only sporozoite antigens will be helping in production of immunity, or only the oocyst antigens will produce the immunity. Other things, other components, those which are present in the merozoites and other things, they will also be useful in development of immunity in the host. Then we also studied the antigenic diversity in various isolates, particularly the allelic or the antigenic diversity was studied in various recombinant proteins that we produced. And we found that these proteins, those we studied, were quite stable, and they were not showing much variations. We produced recombinant proteins, and when we evaluated these recombinant proteins, we found that of all these 7 or 8 recombinant proteins we studied, Eimyria tenella SO7 and Eimyria tenella profilin, they showed the best protection, along with IMP1, immune MAP protein 1. They showed best protection in terms of oocyst reduction. This GAM22, it also helped in reducing the lesion scores and was very significant in terms of weight gain. It was very high, but the oocyst production was not that much reduced in this case. Microneem protein 3 was also tried, and it also resulted in very good weight gain in the bird. We also studied the immune response, and both kinds of immune response were produced. I'm leaving this. Then we also tried with the cocktail immunizations in the birds. We combined all these proteins in different combinations. Not much combinations we could try, but we found that immune map protein, when combined with SO7 antigen and GAM22, it produced the best kind of protection. It resulted in around 95% relative weight gain in birds. 1.67 was lesion score in the birds, infected birds, as compared to more than 3 in the unchallenged group, in the unvaccinated group, and it resulted in around 80% reduction in the oocyst count. So what are the challenges in vaccine development? Though the protective antigens, they can be produced, they are scalable, they can be produced, but it is important to standardize the antigen dose, its regime, the route of administration, the adjuvants. In our studies, we only tried 2 types of adjuvants. We used Titramax Gold and Montanide adjuvant. And even in earlier studies, we used Quint's complete adjuvant and aluminum hydroxide gel also. But what adjuvants are the best, they have to be seen. Then what cocktail immunization pattern should be followed? What proteins they should be mixed? Then another important study that should be done is regarding the cross-protective efficacy. There are reports that some of these antigens, they have a cross-protection potential as well. Those derived from Eimyria tenella can protect against Eimyria acerulina or Eimyria maxima. And the biggest example of this is Eimyria profilins. The profilins, those derived from Eimyria acerulina or Eimyria maxima, they have shown to cross-protect against Eimyria tenella as well. So the nature of immune response is very important, that has to be studied. Then transgenic, another important is, I talked about transgenic Eimyria. You can also use transgenic bacteria and other parasites as delivery systems. So they can also be used. Then fungal systems or plant-based expression systems, they can also be used. A group from Hyderabad, I think Indian Immunological, they express microneem protein in tobacco, and they use it for vaccination of birds in experimental trial, of course, and they were able to show the use of tobacco as a you can say as a delivery system for use of these vaccines. Then after vaccines, the last one I would just briefly touch is the alternate control strategies, which I am not very confident with while dealing with. Alternate control strategies, the first one is the use of probiotics and other feed derivatives, and various bacteria like lactobacillus, enterobacterius, and all these, they have been used. The probiotics basically modify the receptors on the enterocyte. So this will destroy, this will prevent sporozoites or merozoites from penetration. And this will help in destruction of these when they are combined with vaccines. So probiotics, if used with vaccines or with anticoccidials, they are quite effective. And nowadays, people are using. I don't know which probiotic and what. I have listed few of the probiotic compounds that have been tried: fatty acids, vitamins, and direct fed antimicrobials. They have been used, and they are quite effective, as the people say, that if they are combined with the vaccines or with the drugs. Then the market is full of various anticoccidials based on herbals or the natural products or various botanicals. They are either antioxidants or essential oils or immunoactive compounds that have been derived from plants and fungi. And commercially available anticoccidial feed additives are available throughout the world, including India. We have Cocci Zero, Cocci Band, Zyclox that are available in India and others. Then the last is the exploring genetic resistance in the breed. So we can select the breeds, selecting chickens that limit the infectious load either by being refractory to the parasitic infection, that is, we could select resistant breeds, or by tolerating the consequences of infection, which are that is tolerance, or by recovering from pathological consequences of infection significantly quickly to maintain their growth and body weight. That is the resilience. So these three traits are to be looked into, whether these breeds are resistant or whether they are tolerant or resilient. So these are the important things. There are few native chicken breeds globally, one of which is very well characterized is the Egyptian Peomi breed, which tolerates pathological effects of Einidia species. In India, and beside this, there are certain inbred lines of white leghorn chickens, which in which the developmental stages there is variation in the life cycle and the parasite development. And they say that these inbred lines, they can resist coccidiosis to a very good extent. They are available. Then if you see the Indian scenario, in India, though not much work has been done on this, but the people say that a seal breed of chicken, it is comparatively more resistant to coccidiosis, but this has to be proved. I think I have not seen an article regarding this. So this, at the end, I would say that though these are the various control measures that can be adopted, but a single control measure out of this will not help in the control of coccidiosis. So you have to go for an integrated approach, integrating at least biosecurity or the management with the use of anticoccidials and, if possible, vaccines. This should at least be done. Thank you very much for the question. Thank you. I think I'm in time. Yes, almost.
Yeah. Yes, sir. Yes, sir.
Thank you.
So thank you very much for providing such an excellent, enthusiastic, and informative presentation, sir, on this burning topic. Your message was what we want to hear. Touched upon so many critical areas, sir. Thank you, sir, for helping us become more aware about the problems and the ways to solve them in a holistic manner. Now, sir, there are n number of questions.
Yeah, I was expecting.
So, the first question for the most distinguished parasitologist, Dr. P.K. Sanyal, sir. So, the present status of precocious cell lines of Coccidia
in India or globally?
Yes, sir. Global. Maybe.
Let me talk of India first. In India, at present, none of the precocious line is under development at Indian level. A few years back, at least 20 years back, I will say, some work was initiated in Dr. J.R. Rao's laboratory on the development of precocious line of Einidia tenella. After that, as far as my knowledge goes, there is no precocious line that has been developed in India, and that should be developed, I will say, because those are the only feasible methods. Globally, number of vaccines are available on these precocious line, all Hipparacox and LUVAS, Immucox, they are all based on the precocious strains of Einidia. Different kind of vaccines are available from broilers as well as for layers. broilers, we have only four species that in which precocious lines are used, basically Amelia tenella, Amelia tenella, then Acervulina, Maxima, and Nicatrix. These four have been mixed in that are used in broiler industry for precocious lines, and for layer, all seven, the vaccines contain all the seven species of Amelia, the precocious lines for them. Okay, thank you.
So the next question, why it is difficult to distinguish between Amelia tenella and Amelia brunetti, because they are very much site specific. Please elaborate in this aspect.
Yes, but as I showed you in the photograph, in the diagram there, though rectum is involved in Amelia brunetti infection, but in Amelia brunetti infection, some part of Zika or the posterior part of the large intestine is also involved. In Amelia tenella, also Zika and other part of the some part of the large intestine may be involved, and sometimes mixed infections are there, and it becomes really difficult. If it is only the rectal part is involved, then okay, you can say it is Amelia brunetti. But if it is an overlapping infection, then it becomes a little bit difficult sometimes. You will see intestinal coccidiosis in which the whole of the small intestine as well as the large intestine lie right up to the rectum is full of blood. So there will be a mixture of Amelia nicatrix, Amelia tenella, Amelia brunetti, and along with this, who knows, other Amelia species are also present. So you have to actually differentiate them properly.
So the next question is there any national policy of vaccinating backyard chicken against coccidiosis in field condition?
No, big no. There is no national policy till date, and it has to be there.
Okay,
in
sir.
times to come, because no commercial vaccine is indigenously produced, as we say, till date. All these other vaccines that I was telling, they are 2 or 3 that are available in India now. They are being imported only, and they have to take distance.
Okay, sir. Sir, is there an Indian native breed or developed layer line in India to have coccidia resistance?
I told you one they say that it is Asil, it has shown to be resistant, though it is a broiler breed. Then beside this, in Cary, Central Avian Research Institute, they have developed few lines, Cary, I don't remember exactly the name, Cary, some Cary, something is there. They say that it is resistant to a number of diseases, including coccidiosis.
Okay. Sir,
It is not resistant, it is resilient, or you can say resilient, not resistant.
okay. Sir, is there any project working on copro-antigen based detection and differentiation of chicken coccidiosis? Copro-antigen based?
Antigen based? No.
Copro-antigen. Copro-antigen.
As far as my knowledge goes, no. If it is there, I don't know.
Sir, what will you suggest as the most practicable or feasible method of minimizing coccidiosis in backyard farming?
In backyard farming?
Yes, sir.
Okay, in backyard managing, you don't need actually to manage coccidiosis. In backyard farming, how many birds you want to keep, it depends on that. If you are having at least 50 to 100 birds or 200 birds, then try to manage. If it is 10, 20 birds, or like what people keep 20 birds, then management is not that important, because backyard farming is basically on the concept that you will allow the birds to roam freely, they will allow to scavenge freely, and definitely they will, during this process, they will pick up a low level of infection here and there, and they will develop immunity. And the birds that are generally used in backyard farming are the native breeds, and they are comparatively, you can tolerant to Amelia.
Sir, any natural product that has been commercialized for showing high efficacy against coccidiosis in poultry?
I told you the market is full of natural products for use in coccidiosis, and I was reading an article just yesterday. I was seeing people now have started using aloe vera and other things also. And aloe vera has shown to have a very good result against coccidia.
Sir, can dietary changes
Yes.
play a vital role in preventing
Yes.
coccidiosis?
Yes, yes, vitamins, vitamins and minerals, they play very important role. I told you in the probiotics and where they have shown vitamin A, vitamin K, and zinc, they have a very vital role in coccidiosis.
Sir, what are the side effects of coccidiostats in chicken feeder or that is consumers?
Side effects, as such, there will be now consumers are very wary of residues, drug residues. So those drug residues may be carried over to the humans as well. But I told you in coccidiosis, the coccidiostats and the anticoccidials that are used, the withdrawal period is not very long. It is 1 to 7 days only. So if you are using coccidiostat, and if you want to market your birds at say around 8 weeks of age, then stop using coccidiostat at up to 7 weeks of age. If you will stop at 7 weeks of age, by the market, when you want to market it at 8 weeks or 7 weeks, the residual effect will have been reduced.
Sir, this is question from Dr. Saad.
Yeah.
Turmeric and ginger have any effect against coccidiosis in both chicken and small ruminants? Sir, do you have any experience in using them?
No, I have never used any of the herbals for any of the parasitic diseases. But yes, they have.
And the same question also, sir, is there any impact of dietary supplementation of flaxseed?
Yes, people have worked on flaxseed. Yes.
Sir, another interesting question, sir. How do you identify B5 homozygous chicken who are more resistant to coccidiosis?
Yes, people, they have been working on these MHC genes for identifying the species. So they have this is SNP based typing is being done by people, some of the groups throughout the world. Generally, this kind of work is done by genetics. Genetics people are using this kind of approach.
Okay, sir. Sir, whether infection and treatment policy can be applied here as it is applied in most of the hemoprodrogen diseases.
What is vaccine and treatment policy here that I propose that I showed? Vaccine live doses are being given, and actually then this is followed by treatment with the anticoccidials. So it is an infection and treatment policy in one way. Infection in very low doses to develop immunity and then treat it.
So sorry, sir, there are 2 to 3 more questions. Actually,
no
highly enthusiastic. So last 2 to 3 questions, sir.
Okay.
Which factor is more responsible for higher incidence of coccidiosis? Is it a management factor or climatic factor?
Management. The prime factor is the management. If you are managing it properly, whatever the climate will be, you can manage it. If it is, even if it is rainy season, you are able to keep your litter dry, you will be able to manage coccidiosis.
Yes. The last question is, sir, the last two, is it beneficial to give coccidiostat at preventive doses if there is a history of incidence of coccidiosis in the previous batch?
Yes, coccidiostats are routinely used. There will be hardly any flock or any owner who will not use coccidiostat. All veterinarians, everyone will recommend coccidiostats. If there is coccidiosis or if there is no coccidiosis, if it was there in previous batch, then what they do is they will first clean up the infection. Cleaning up the infection means thorough cleaning of the farm, sheds, birds, everything, and then resting the shed and then in new flock and using the coccidiostat. You may shift the coccidiostat in such a case. Class of.
Okay, sir. Thank you very much for giving such an excellent presentation, sir. And I will request Dr. Snail to please propose vote of thanks.
A very good afternoon, everyone. It gives me immense pleasure that I have been assigned the job to deliver the hearty vote of thanks to Dr. Rajat, who has been my mentor during the PhD program in the IVRA. He is a jolly person, a cheerful personality, and a great teacher and a simple human being. Sir, all these qualities were very well reflected in your presentation. Coccidiosis is an inevitable problem in the poultry industry, and your constant effort to find out a plausible solution in the form of a vaccine is remarkable. Your presentation gives us inspiration that a researcher should be well focused and strive hard to ultimately reach some outcome. For us, you are a perfect researcher. At this note, I would like to quote some words of Dr. APJ Abdul Kalam, profession that shape the character, caliber, and the future of an individual. If your students remember you as a teacher, that will be the biggest for any teacher. Thank you, sir.
No doubt about it.
At this end, on my own behalf and on behalf of the organizing committee, I extend my heartiest gratitude to you, sir, for sparing your valuable time and sharing the lead in the poultry coccidiosis with the budding veterinarians. Thank you once again.
Thank you, sir. Mr. Neil. Thank you.
Now I request the organizing secretary, Dr. Bora, to share some words and release the e-compendium for the participants.
Good afternoon, everybody. On the behalf of organizing committee, I pay my sincere gratitude to all the senior teachers, speakers, and thanks to the participants who all have gathered on this virtual platform to make this webinar possible and successful. During these 3 days, the participants of webinar series were together for receiving the knowledge from experts and answer to the queries of the problems we were facing during their research work. The information has updated the knowledge of all the listeners and participants. Once again, I thank the experts for their lectures which they have prepared after years of the groundwork and experience they have gained. I also thank the stalwarts for their video messages. Now, I, along with my committee members, release the e-compendium of around 110 pages. This is of around 110 pages of e-compendium, and this e-compendium, along with the e-certificate, will be provided to all the active participants who were there during the 3 days of webinar series. At the last, now I invite Dr. Snehal Gupta to propose the final things or vote of things. Thank you.
Now the time has arrived to give faith to all the participants and to show our gratitudes towards all those who have contributed to make this event a great success. First and the foremost, I would like to thank our worthy Vice Chancellor, Lala Lajpat Rai University of Veterinary and Animal Science, Dr. Gurdial Singh Sir, for providing us courage and facilities to organize such an event during the COVID pandemic. We are highly grateful to our patrons, Dr. D.P. Sharma and Dr. S.K. Gupta, for their word of encouragement and constant guidance during the webinar series. Our heartfelt thanks to the executive member of our IAAVP Society for their valuable contribution, wider publicity, and consistent support in all our efforts. Also offer a special gratitude to our technical committee, Dr. Nilesh Sharma, Dr. Tarun Kumar, Dr. Jadhav, and Dr. Sumit Mahajan, and our media partner who have worked hard to ensure that the Parasitology web series become a memorable success. I feel immense pleasure while announcing that around 1,100 scholars registered for our webinar series. We all are honored to have such an enthusiastic and motivating audience. I am so grateful that scholars from other countries like Spain, Canada, USA, Pakistan, Bangladesh, and Nigeria also participated in the webinar series. We wholeheartedly thanks our distinguished speakers for accepting our invitation and sharing their lead in the subject with the young generation. From the life journey of our eminent speaker, it was quite evident that hard work always pays off. Practice and the consistency is the key to perfection. Patience and discipline take you to the next level. Staying alert, strong, fair, and focused get us closer to our goals. During the 3 days of the National Web Series, our eminent speakers, Dr. Sanjay, come up with several new diagnostic and therapeutic approaches in the equine pyroplasmosis. Dr. Gatne is an internationally renowned canine practitioner and also credited for the discovery of new hookworm species. Dr. Shri Kumar took the wildlife parasitology to a next level. Dr. Rajat Garg is persistently working on the diagnostic and the development of vaccine against poultry coccidiosis. And last but not the least, Dr. Singla and the Dr. Rajesh, with their wide teaching experience, took us to a new horizon of the veterinary parasitology. At the end, I would like to say all our reserved and unreserved appreciation goes to the Almighty God for making the event a great success. We are indeed grateful to all those who directly or indirectly supported us in making the webinar a grand success. Finally, I leave you all with the inspiring quotation amidst of this corona. At times when our light goes up and is rekindled by a spark from another person, each of us has cause to think with our deepest gratitude of those who have lighted the flame within us. So we all are indebted to our speakers who ignited the flame of knowledge gained within us. Thank you once again, all the speaker participants and everyone who has contributed to make this event a grand success. Thank you, everyone. Important notice for the participant: compendium and the e-certificate will be issued to all the registered participants within the 10 to 15 working days. Thank you all.